The proper reading frame is going to determine whether or not the protein that is ultimately made from the mRNA sequence is going to be correct and whether that protein is ultimately going to work properly inside of the cell. So if we talk a little bit about the reading frame is really the proper groupings of 3 nucleotides. So really getting the groups of 3 correct, and if the groups of 3 are correct, then the amino acids that are incorporated will be correct. So as an illustration, if you look at the sentence right here, the top sentence, the red dog ate the cat, that sentence has some meaning. All of the words are made up of 3 letters each, and we read them in those specific 3-letter combinations, and it gives us some meaning. Now, if we just shift the 3-letter combinations over just one, or over 2 letters, as you can see in the bottom one, notice that the groups of 3 start to make completely different sense, or really in this case, they don't make any sense at all. And that is a shift in the reading frame. So we have changed the groups of 3, and now it has different meaning than what it had before. And when we're looking at the DNA, it was really exactly the same thing. If we look at what we have right here now, this is a DNA sequence, and you can see that there are 3 different reading frames being shown here, and each of the reading frames is ultimately going to result in different amino acids being incorporated into the protein. So we do have to make sure when the ribosome is beginning the translation process that it does choose the correct group of 3 to start with, because once it chooses the first group of 3, all of the rest of those are going to fall into place afterwards. So the correct group of 3 will set the reading frame for the entire polypeptide that we are ultimately making.